Hello, students. Welcome back to another session. Today, we will be discussing the another topic, biotechnology, in that recombinant DNA technology principles and processes. So, let's start. What is biotechnology? It is a part of the modern biology where we are using some living organisms or a particular cells. Okay, suppose the microbial cells, microorganisms or plant cells, plant cell or animal cell. So with the help of those cells or the cell components, we are making enzymes or biological products, and those products will be useful to humans. So with the help of technology and integrated with the natural science or the living organisms, we are producing the products which are useful to human. That's what biotechnology. Okay, so biotechnology, there are a lot of applications in different field. For example, in the medical field, agriculture, environment, various industries, biotechnology has a crucial role nowadays. So this chapter we will be discussing about what are the different principles and processes under the recombinant DNA technology. Okay, so when you compare the traditional technology and the modern biotechnology, traditional technologies, there was like we are using various technologies in which we are using the microorganisms like a fermentation of bread and alcohol. There we use that yeast and all, and dairy products, cheese, yogurt, other microorganisms we are using, right? Then the traditional brewing, wine making also, there's Saccharomyces cerevisiae, food preservation techniques, we use some chemicals. All those are related some microbes or some chemicals we use for making the products or preserving the food items and all. But in modern biotechnology, this technique, we are going to the molecular level. Okay, molecular level means we are manipulating the genes. Okay, genetic manipulation will be done, which result in the production of certain products which are very useful to humans. So this was emerged in the late 20th century. So this biotechnology, there we will discuss about the genetic engineering, DNA cloning, recombinant DNA technology, tissue culture, cell biology, protein engineering, and protein synthesis. All those are involved in the modern biotechnology. Okay, so when we think about the applications of biotechnology, biotechnology are very useful in the agricultural field, in the medicines, in the environment, in the various industries. Coming to the agricultural field, earlier farmers were using the fertilizers, manures, and all for getting the good produce from the plants. But plants, there are a lot of factors that affect the growth of the plants. For example, the pest or weeds and all, right? So then they were thinking about instead of using the chemicals, how can we change the genes of the plant so that it has some resistant power, or it will be resistant against the pesticides, pest and all. Okay, so in that way, through the biotechnology, scientists modified the genes of certain plants. One such example is the bt cotton. It's an insect resistant plant. Then another one is a golden rice. Golden rice, we there is a specific point. It has vitamin A is there. Okay, so along with the other nutrients, carbohydrate and all, they have a vitamin A is also additional nutrient is there. So herbicide tolerant soya bean, disease resistant varieties of plants. So these are some of the examples of applications of biotechnology in the agriculture. Coming to the medicine, there are a lot of uses there. For example, diagnostic tools are there through the biotechnology, and also the medicines related to this. Human insulin now developed from bacteria. Okay, and the vaccines, recombinant vaccines are produced, monoclonal antibodies for diagnostic, and gene therapy is there. All these are come under the medicines, right? Then coming to the environment. In the environmental biotechnology, mainly that deals with the bioremediation of contaminated soil, removing the microbes and disease causing organisms from the soil, sewage treatment plants, spill cleanup, air pollution control, all this are coming under this environment. And industries, enzymes production, biofuel production, biodegradable plastics, food processing aids, and all some examples of the applications in the industry. So, biotechnology is very, very essential in now the modern biology, because there are a lot of problems can be solved by using biotechnology, right? So let's start the main topic, principles of biotechnology. So in the principles of biotechnology, when we are thinking, there are two core principles are there in biotechnology. One is the genetic engineering and the other one is the chemical engineering. So in the genetic engineering, we are changing the genetic structure of the organism. Okay, so we are altering the genetic material of the organism. That means we are introducing some desired characters into it by cutting, then modifying, inserting the genes into the organism so that we will get some desirable characters. Such kind of organism, we are using the term GMO. What is the full form of GMO? What is the full form of GMO? It is genetically genetically modified organism. Genetically modified organism. Okay. So in genetic engineering, that is mainly deals with the production of genetically modified organism by altering the genetic structure of the organism, right? And in the second core principle, maintaining pure culture, or in the chemical engineering, that means mainly deals with the maintenance of sterile and or contamination free culture of host organism in controlled environment for a large scale production of desired products. Okay, so these are the two core principles of biotechnology, genetic engineering and chemical engineering. So these principles work together, which result in the production of desirable products, right? So in the biotechnology, when you are going to the actual topic, we have to learn some terms. Okay. So first, as I already said, it is genetically modified organism. Another term you need to remember, that is vector. Have you heard about vector in the previous chapter, human health and diseases? In that, what is vector? Vector is a carrier of disease causing microbes. Okay. Here in biotechnology, vector is also a carrier. It's a carrier. But what does it carry? It carries specific gene. Okay. That gene is called a gene of interest. Okay. So the vectors are the carrier of gene of interest. Then what is the gene of interest? Of interest. A desired gene. Desired gene. Okay. The gene with the desired characters. Gene with the desired characters. Gene with the desired characters are called a desirable gene or a desired gene or gene of interest. Okay, so genetically modified organism, vectors are the carriers of gene of interest. Gene of interest are the gene with which carry the specific characters or desired characters. Right? Another term is host organism. Host organism, they are the organisms which carry the gene of interest. Okay, so organisms carry the gene of interest. For example, bacteria, E. coli, Escherichia coli is an example of a host which carries specific gene of interest. Then another specific term you need to remember, that vector, we know what is a vector. It is a carrier of gene of interest. Okay, in that one vector is called a commonly used vector is known as plasmid. And about the plasmid, you might be learned in the structure of the bacteria because the plasmid is present in the bacteria. So plasmids are bacterial extra chromosomal DNA. Extra chromosomal DNA. And this plasmid can be used as a vector for carrying the gene of interest. So remember these different terms: vectors, gene of interest, host, plasmid. Two more are there. They are like a molecular scissor. Okay. One is the molecular scissors. Molecular scissors. Another one is known as the molecular glue. Molecular glue. Molecular scissors and molecular glues both are the enzymes. Okay. So one enzyme is called a restriction enzyme, which are known as the molecular scissors. Restriction enzymes. Restriction enzymes are called molecular scissors, and molecular glue is the DNA ligase. DNA ligase involved in the ligation of the DNA, joining of the DNA. Okay, so this is joining. This enzyme help in the joining of the DNA. So what will be the function of the molecular scissors? Molecular scissors function is to cut the DNA. Okay, so cut the DNA. So in this important terms, remember GMO, vector, gene of interest, host, plasmid, molecular scissors, and molecular glue. Now let's start with the recombinant DNA technology. Okay, so what do you mean by a recombinant DNA or rDNA? The DNA with some extra specific genes, they are recombinant one, then we can call such DNA as the recombinant DNA. So the first recombinant DNA was constructed by two scientists in 1972, Herbert Boyer and Stanley Cohen. Very, very important. Okay. So 1972, Herbert Boyer and Stanley Cohen, they constructed the first rDNA. Okay, so how they constructed and what are the materials required for those construction of the first rDNA, that is important point we need to discuss. So the key components, should be the 4 in DNA, there is a gene of interest, and they have taken the gene of interest, that is the antibiotic resistant gene. The gene which help to resistant against the antibiotics. That is the gene they selected. After that, they want to carry those genes into the host body. So, who is a carrier? The vector is the carrier of the gene of interest. And in this experiment, Stanley Cohen and Herbert Boyer, they took plasmid from the bacteria Salmonella typhimurium as the vector. Okay, so remember the gene of interest here is the antibiotic resistant gene. The vector used as a plasmid from the bacteria Salmonella typhimurium. Okay, and then with the help of, so plasmid is a circular DNA with the help of specific, so suppose this is the gene of interest. Okay, and with the help of molecular scissors they cut the gene of interest. What is the molecular? Is a restriction enzyme. Okay, with the help of restriction enzyme, they cut the gene of interest, and they have taken another bacteria plasmid. Okay, that bacteria is the host organism. So, what is host organism? The organisms which carry the gene of interest. So, here the host organism is the Escherichia coli, E. coli. Okay, so first they isolated the gene from the Salmonella typhimurium and then inserted the plasmid of the E. coli. E. coli bacteria into the gene of interest. Okay, the gene of interest they inserted. Insertion, the joining is with the help of which enzyme? DNA ligase. Okay, so they cut the DNA part and then joined the extra gene. And after that, they inserted this plasmid into the E. coli bacteria. Now, in the E. coli bacteria, that specific gene of interest is there. Every 20 minutes, it multiplies. Along with its cellular components, this gene of interest also multiplied. That means they got number of copies of the specific gene. Okay, so recombinant DNA technology, remember the three key points: cut, copy, cut, paste, and copy. Computers, we'll study about cut, paste, and copy. Okay, the same thing in recombinant DNA technology. We are cutting the specific gene of interest, then pasting into the vector, and then we are copying in host body. Right? So, recombinant DNA technology, there are specific tools required. We have to learn more in detail about the specific tools. Okay, so the steps involved in the DNA technology. First, we have to identify the DNA. Okay, that is, we are just recapping once again. We need to identify the DNA, where it is located, which is the specific gene of interest, everything. First we have to identify, then cut the gene of interest and introducing into the host body, transferring the identified DNA or the gene of interest into a specific host through the vector, and maintain and transfer those DNA. These are the fundamental steps for making the rDNA. Okay, so let's start with the tools of recombinant DNA technology. The very first tool, remember, a restriction enzyme. Okay, so we already know that restriction enzymes are called molecular scissors. Why are they called so? Because they cut the specific part of the DNA. Okay, so they will find out the specific region of the DNA and they cut the specific that region. That is one tool. Another tool is the vector. What is the vector? They are the carriers of the gene of interest or the foreign gene into the host body. Okay, so we can call these are the vehicle. In biotechnology, the vehicle or the carriers of the gene are the vectors. Then another tool is the host organism. So some living cell is required to receive the recombinant DNA. Okay, so those host organism act like a biological factories for the protein production. In addition to these three different tools, there are certain enzymes also required. For example, DNA ligase. I have told you, DNA ligase, what is another name? They are called a molecular glue. Okay, because they are involved in the ligation or the joining. Right? And then there are polymerase enzymes there. Polymerase enzymes, we have learned in the previous chapter that they are involved in the replication of the DNA. Okay, so each tool has a specific role in the rDNA technology. Let's start with the first tool, that is the restriction enzyme. So, a restriction enzyme, for how where it found first. Okay, so restriction enzyme are naturally present in the prokaryotes. specifically in the bacteria as a defense mechanism. So, in bacteria, there is a defense mechanism to inactivate or to kill the bacteriophage. We know what are bacteriophage. Bacteriophages are the virus which infect the bacteria, right? So we know the structure of the bacteria. Bacteriophage. They have a protein coat is there. This is the protein coat with the protein coat fiber is there, and inside you can see the DNA. Okay, the only very simple structure. So this is a bacteriophage. So when the bacteriophage infecting the bacteria, bacteria has certain specific defense mechanism. So they have a what scientists found that in this bacteria they have two enzymes there. One enzyme is to add the methyl group into the DNA. Okay, methyl group into the DNA of the bacteriophage. Another enzyme is to cut the DNA of the bacteriophage. Okay, so the second enzyme that is called a restriction enzyme. Okay, so in their 1963, two enzymes responsible for restricting the growth of the bacteriophage in E. coli were isolated. One will be adding the methyl group in the DNA of the bacteriophage, while the other enzyme is used for cutting the DNA. And those the enzyme which used to cut the DNA, those enzymes are called a restriction endonuclease. So restriction enzymes or the restriction endonuclease are the molecular scissors. Later on, they scientists used this enzyme as the molecular scissors in biotechnology. Okay, so what are restriction enzymes? They belong to the class nuclease. We know the nuclease enzymes, they are involved in the digestion of the nuclear components. So this restriction nuclease or the restriction enzymes, they are of two different types. One is called exonuclease and another one is the endonuclease. Exonuclease will cut the outer part of the DNA. Like this, they will cut the outer part of the DNA. Okay. So those enzymes are the exonuclease. Endonuclease will cut the specific part inside, the deep inside part of the DNA. The exonuclease remove the nucleotides from the end of the DNA, whereas the endonuclease cut the specific position within the DNA. That is clear now. Distinguish between exonuclease and endonuclease. So this restriction enzymes, they have a specific features. They just go through the DNA and they will find out specific sequences, and those sequences are called recognition sequence or palindromic sequence. What are palindromes? When we are reading from from the right to left and left to right, it will be same only. For example, I'll write Malayalam. When you are reading this from this side or both the sides, it will be same only. That is the palindrome. So this restriction enzyme, they recognize certain sequences in the DNA. They are the palindromic sequences only. Okay, and we will come to that part again. Palindromic sequences, and they have a specific recognition site. There are different types of recognition site. Okay, nowadays scientists found around 600 above restriction site from 230 strains of bacteria. And all those different kinds of restriction enzymes, they have a specific recognition site. And those enzymes are very necessary for DNA cloning or the multiplication of the genes. And there is a specific name for each restriction enzymes that is based on the bacteria in which they are taken. Right? So let's discuss about the nomenclature of the restriction enzyme. Nomenclature, when we are writing, when we are giving the name of the restriction enzyme, one restriction enzyme, we can say EcoR1. Okay, this is one restriction enzyme, EcoR1. This is not the bacteria, this is the restriction enzyme. So when you are giving the name, the first letter should be the genus of the bacteria. So genus, when we are considering the genus, the first letter of the genus, we are using the first part. Okay, so here the genus first letter. So what is the genus of the bacteria? Escherichia. In that Escherichia, the E will be taken here. And second and third one. Second and third letter will be the species two letters, first two letters. Species, first two letters. So what is the species? Coli. So from this, E first taken, then the next two letters go, right? And the fourth one, R, is the strain in which bacterial strain in which this one belong to. So R strain bacteria. Okay. And this is not I, this is the Roman numeral. This Roman numeral indicate the order in which the bacteria isolated. Right? So this is the nomenclature. We are giving the name of the various restriction enzyme based on the source of bacteria. Okay, so just once again, restriction enzymes named systematically based on their bacterial origin, reflecting the genus, species, strain, and order of discovery. Example here, the first letter, the genus of the bacteria, Escherichia coli, in that E is there. Then the next two letters, species of the bacteria, Co. Fourth letter, strain of the bacteria, R strain, RY13 strain, okay. And the Roman numeral, the order in which the discovery. The first enzyme discovered from this strain is the EcoR1. That's why the name, okay. Clear? The nomenclature or the naming of restriction enzyme. There are around more than 600 restriction enzymes, different names there. Okay, so till here we will be winding today's class. We will be continuing in the next class regarding more features of the restriction enzymes. Till here, if you have any doubt, you can ask now. Okay, so no doubt, right? Thank you, all of you.